So good morning, everyone, and thank you so much for joining me today as I present my thesis on using environmental DNA on examining marine finfish biodiversity in Arrábida Marine Park, Portugal. So as more species face increasing risk to extinction due to habitat loss caused by overfishing, coastal development, pollution, and climate change, the need for biodiversity monitoring is essential to predict ecosystem responses. In this way, predictions can lead to better management and conservation interventions, especially to those that are applied to biodiversity hotspots. So one such place is the Arrábida Marine Park, located in the western coast of Portugal. This is a 38-kilometer coastline and is a Natura 2000 site, and it's a very important transition zone in the Lusitanian biogeographic province, as it harbors more than 2,000 marine flora and fauna from the Mediterranean, subtropical, and northern temperate countries. The high species richness and abundance in the park make it a very valuable area for fisheries. However, the previous high fishing pressure and recreational nautical activities in the park before the park was established have destroyed certain habitats in there. So as part of the management and conservation efforts of the park, restoration activities have been established to improve the conditions of these habitats, especially for its marine vegetations. So commonly used in biodiversity assessments, the richness and abundance of finfishes such as bonafishes and elasmobranchs have been used as indicators of marine health, as these organisms have various ecosystem roles and services which includes but not entirely limited to resilience and fisheries. So any change in the assemblage of these species may demonstrate how disturbances or interventions have affected the system. So increasingly used in recent years, the use of environmental DNA or free genetic DNA from cells released through the skin or gametes or any decaying matter have been revolutionary in estimating and assessing marine biodiversity. So eDNA metabarcoding is non-invasive, as it uses environmental samples such as seawater and sediments, and it is cost and time efficient compared to conventional and traditional biodiversity approaches, as it is as next generation sequencing approaches or techniques have advanced and become more affordable. Thus, the objective of the study is to examine marine finfish biodiversity in natural and restored marine vegetations, such as seagrass meadows, Schistosera beds, and kelp forests in Arrábida Marine Park, using this relatively new approach to marine biodiversity. So using eDNA metabarcoding, we wanted to demonstrate patterns in species richness, abundance, and assemblage by using a recently published 12S rRNA MyFish primer together with Illumina MySeq next generation platform. So to do this, we did a two-way approach. So we collected water samples, three replicate samples per site using scuba diving or free diving. And then we extracted the eDNA from the samples by manually filtering them using Sterevex capsule filters. And since the eDNA are very minute in the seawater, we did a two-step PCR approach for the amplification using the MyFish primer. So before we proceeded to downstream analysis of our samples, we first built a custom reference database by downloading 12S and My2 genome sequences from NCBI as of November 2020 for fish and elasmobranch species. So we assembled these sequences and then filtered them out and quality assessed them using our quality assessment criteria, and then filtered those that do not satisfy these criteria. From there, we identify the species that are present in the park but do not have the target MyFish barcode, and then did DNA barcoding using fin clips and Sanger sequencing. So from the NCBI sequences and the newly barcoded sequences, these sequences were then fed into the ANACAPA toolkit, which is a recently developed modular toolkit intended to accurately and easily customize bioinformatics pipelines for user-specific amplification of eDNA. So we used the first module of the ANACAPA to customize our Arrábida reference database to include their pre-built 12S MyFish library to enhance sequence coverage, thus creating a more comprehensive reference database. We then proceeded to using the module 2 after we obtained the raw reads from our MySeq sequencing. So this second module does standard DNA quality control and generation of amplicon sequence variants using CutAdapt and Dada2 tools. Afterwards, we use the third module, which uses both Bowtie2 and Bayesian lowest common ancestor method that assigns taxonomy to our ASVs by determining the lowest common ancestor from the multiple weighted hits. And afterwards, lastly, we run the process data through Rana Kappa, which does data visualization and exploration, and as well as standard biodiversity statistics, including accumulation curves and diversity indices, among others. So this package utilizes vegan, phyloseq, and the ggplots of R. Unfortunately, due to the restrictions caused by the pandemic, not all samples were analyzed in time, and so I am only able to present the results we have for the seagrass meadows. So results show that we have an increasing trend for the accumulation curves, and this indicates that with more sampling, we will be detecting more species. And as you can see, the curves do not seem to reach plateau, and this as well indicates that with more sampling effort, more species can be detected. Furthermore, we also saw that the Shannon's index was relatively higher in the natural seagrass meadow, but this is not significantly different with the restored site. And this, we can infer that maybe the sites could have or harbor similar assemblage, but with varying species abundance. And this was actually we can actually see this in this figure. So the mean read count, although not significantly different, was higher in the restored seagrass, but compared to the natural seagrass. So in this Venn diagram, the size of the circle corresponds to the mean read count, and the font size of the species name actually corresponds to the percent relative composition of the species. The arrows corresponds to which species have more than 1,000 reads, and the orientation of the arrows corresponds to which site do they appear with higher read count. So again, the mean read count was higher in the restored site, with Pagellos bogaraveo comprising more than 50% of the total reads. So more than 60%, or 15 out of the 24 species that were detected, were found in both sites. And from the 24, 2 species are in the IUCN Red List, namely the Pagellos bogaraveo, or the black spot sea bream, under the near threatened category and was found in both sites, while the Trachurus or Atlantic horse mackerel, which is under the vulnerable category, was only found in the natural seagrass meadow. So you can also note, noted in asterisk, we also found 20 commercially important species, most of which were detected in the natural seagrass meadow. So these results show that read count could be an indication that more individuals or bigger individuals could be found in the restored site compared to the natural seagrass meadow. So the presence of more species in the natural seagrass meadow may be an attribution of a larger patch in there, thus a more bigger or has a more complex habitat configuration that can support more biodiversity. So this is because habitat configuration and complexity influences food, habitat, and protection, thus ultimately affecting abundance, diversity, and distribution of your species. Conversely, the high read count also, which could be correlated to species abundance or biomass, could be an indication, could be due to the partial protection of the restored site as it is located in the fisheries regulated zone. So this is consistent with studies that have found higher read count in protected or remote areas. So this means that more individuals or bigger individuals could be expected to leave more eDNA in areas with minimal to no anthropogenic threats. So we also saw overlapping species assemblage between the sites, and we somehow linked this to the restoration could be a potential restoration benefit, as the restored site may have developed the same complexity that was found in the natural site, thus being able to support the same assemblage as those in the natural site. Or this could also be a case of connectivity, as the sites are approximately 6 kilometers from each other. So oceanographic factors such as waves and currents may have facilitated an exchange of eDNA or DNA between the sites. So absence of elasmobranchs such as sharks and rays could be attributed to very low eDNA traces, as these species are very transient visitors in this area that forage in this habitat but use other habitats for foraging and reproduction and other things. So, but this could also be a case of primer sensitivity, such that several studies have highlighted the non-detection and low resolution of some eDNA primer, including the myfish for certain taxa and for closely related species. But even though, although this is the case, the species accumulation curves could have indicated that with more sampling, more species could be detected, and with the use of a more specific primer, the total biodiversity could be enhanced, which could include rare species such as elasmobranchs. So to summarize the results of this study, so eDNA metabarcoding has showed the differences in biodiversity between the restored and natural seagrass sites for marine finfishes, and these observed differences could be driven by availability of the large and complex habitats, as well as the protection and regulation of fishing in the fisheries regulated zone. So the benefits of management and conservation activities may have been shown in the overlapping species assemblage as an effect of the recovery from previous disturbances or the connectivity between the sites. The presence of IUCN and red list and commercially important species could be also derived benefits from these efforts. So these results highlight the applicability of the technique to document and examine patterns of marine finfish biodiversity and its potential use as a complementary biodiversity tool to monitor the park. And in the long run, this tool could also be adapted as a supplementary tool to traditional biodiversity methods that are often invasive, expensive, and laborious. So this work would not have been possible without the support and guidance of these people. So Dr. Anna, Dr. Garrett, Dr. Esther, Luca, and to ISPA, especially to Dr. Joanna Robalo, who have been very instrumental in improving our Arabida reference database, and to our funding agencies and partners, the ICNF and Info Barimaris project. Thank you all so much, and daghang salamat and good day.